I want to be able to tell you a story that has been pretty recently published from the lab of Waxman that I was part of, and it'll be aspirational for the idea that we can try to predict the responsiveness of patients to drugs. And so, with further ado, which way? No, that way. Um, since this is a 9-channel symposium, I decided not to go in depth into the background of sodium channels, voltage-gated sodium channels, just pointing out that peripheral sensory neurons have specific voltage-gated sodium channels that are highly expressed, and so they are of high interest in the regulation of sensory signaling and in particular pain. And the isoform that the Waxman lab has been studying for quite some time has been NAV 1.7, and it's become a highly validated target. All 3 isoforms, however, are linked to pain in patients that carry mutations. Also, drug development targeting the NAV channels is ongoing, but a good one has yet to be described. I'll also talk to you about pain conditions such as inherited erythromelalgia, the IEM, or particular, for this story, small fiber neuropathy. And both of these pain conditions have a component of mutant or variant sodium channels, and they often have poor or variable responses. So I'll be telling a story that looks at this variability. So small fiber neuropathy is excruciating burning pain in the hands and feet. Most people, or less than half of the patients, find relief with current compounds, and usually the reason they don't get enough relief is that the compounds concentration that's required also starts eliciting side effects. In addition, as I mentioned, that some patients do respond reasonably well, but whereas others don't. And so this study is attempting to address that. And I'm going to be telling you how a study was performed using Lacosamide. It's one of the family of anti-epileptic drugs, so it's already approved for epilepsy. But because of mechanisms of action targeting sodium channels, it's something that people who study pain can try and document whether they're going to work. And so this is all about, can we discern the discrepancies or the differences and thus lead to better treatments for these patients? What I'm showing here is that the mechanism of action of lacosamide is somewhat distinct compared to other anti-epileptic drugs, lidocaine or carbamazepine, in that the primary biophysical signature that gets altered when lacosamide binds is that it enhances slow inactivation. So, oops, and I moved it. Sorry, figuring this out. All right, so lacosamide prime signature is if you do a protocol that elicits slow inactivation, so these are long conditioning pulses, you get a shift to the left in the voltage so that at a particular holding potential or resting membrane potential, the availability of the channels is decreased. And that's its primary mechanism of action. And not shown on this is lacosamide also has a very slow kinetics of onset or offset. So it's also distinctive compared to lidocaine or some of the others. And so this is just 2 studies where I tried to show that there's the main effect on slow inactivation and barely any effect on fast if you do the protocol for fast inactivation. There is some results in the literature that argue whether it's specifically binding to the slow inactivated state or it's just slow kinetics binding to the fast inactivated state. But for my purposes, I am following the signature. Does it shift when I do the protocol for slow inactivation, or does it shift when you do a protocol for fast inactivation? So the story really starts with a long-standing collaboration we have with a group in Maastricht, the Netherlands, and they are experts at characterizing small fiber neuropathy. And as part of our collaboration, they became very interested to evaluate are sodium channel variants underlying small fiber neuropathy. Now, small fiber neuropathy can be caused by conditions such as diabetes or exposure to toxins, but there is a significant fraction of patients who present with small fiber neuropathy who don't have a predisposing event. So they're idiopathic. You don't understand why. And so the first study they did was they took a whole bunch of these small fiber neuropathy patients who were idiopathic for known reasons and decided to do sequencing of the ion channels that are known to be regulating peripheral nerve excitability. And in particular, we're focused on NAV 1.7. And so this just goes down and shows that of this number, about 11% were found to have variants, differences from wild type channels. And so, this is potentially really exciting. There's a reason for them to have the pain condition. And since the reason is a sodium channel, if you could target the sodium channel with a pharmaceutical, hopefully that would help them with their pain. So they did this trial, and they used Lacosamide for a couple of reasons. Lacosamide was already reported to have less severe side effects than some of the other anti-epileptic drugs. But in addition, during our collaboration, when we worked up the first few variants that are linked to small fiber neuropathy, and there are a variety of gain of function changes in their biophysics. But one of the things that was consistent or common among all the small fiber neuropathy variants is that they all impaired slow inactivation. So the channels were slower to be inactivated by depolarization. And since Lacosamide it appears to be selectively targeting the slow inactivation process, it was chosen to be the investigational drug. The study design was a crossover, and I forget the total number. I guess it's 25 patients, all of which carry some variant in 1.7 were enrolled, and the trial was a success. Statistically, there was an effect of Lacosamide to improve the pain. When the study was first published, it was just pooled. But because we have this very long-standing collaboration, they allowed us to break the code. So when you look at different patients, some of them responded well and others didn't respond so well. And so we broke out and characterized the Lacosamide effects of 5 variants, mostly because we have variants where we have multiple patients so that the statistics are good, and others because they were either sort of significantly non-responsive or significantly responsive. So the next slide is, and when we did this study, we chose to use a concentration of Lacosamide that's clinically achievable, in case part of the reason is maybe the IC50 was shifted. Anyways, I will show a very busy slide now, and I'll try to walk you through it. Where so here's our mutants that are in columns, and the top row is the fast inactivation characterization in the presence or absence of the lacosamide. Absence is black and lacosamide is red. The second row is slow inactivation, and the bottom row is use dependence. And you probably have already detected things that look different depending on whether we scored it as a responder or a non-responder. And so I'll leave the boxes up. You can continue to look at it and see if you can detect things that appear to be different for responders than non-responders. One of the things that we really picked up on is in the upper row, as I said, lacosamide's not very effective at modulating fast inactivation, but in our responding variants, we now have a very clear modulation of fast inactivation compared to, these kind of effects are already what you expect to see, even if it was a wild type channel, which is in the paper, but I just don't have it on this thing. You can make the case that slow inactivation is also enhanced by lacosamide in responders, perhaps in the bottom part, the fraction that's unable to be slow inactivated. So, normally it's like 20% is always available no matter how long, but it goes down to 5 or 6. And then maybe use dependence is enhanced also. But I do want to spend the next few slides to show our efforts to understand this variant that was profoundly non-responsive. I mean, it doesn't change at all to any of these protocols. And part of the other reason we're interested in this variant is that it helps us to address, well, where is or are the sites of lacosamide action? If lacosamide is an anticonvulsant, it should go to the local anesthetic site, which is in the vestibule of the pore ion permeation pathway. But the W1538 residue is in this novel inhibitor interaction site in the extracellular facing end of the voltage sensor domain of domain 4. And it's actually the M2 residue. So this very groundbreaking paper that showed that you could now develop compounds that had specificity between at least families of the channels. So in this paper, they talked about a compound that was very effectively blocking 1,3 and essentially had no effect on 1,7. And this entire shift between the 1,3 phenotype and the 1,7 phenotype was determined by 3 residues that are different between 1,3 and 1,7. And it turns out this residue here is the one that we're interested in. So it's part of this binding site. We've performed an in silico docking study to see where lacosamide is binding. And so this is a recently published cryo-EM structure of the human 1,7, and I've taken the side view and the top view, and the W1538 site is shown here in yellow, and I've also put in cyan, there's 2 amino acids that can disrupt the local anesthetic site. Next, I zoom into either here or here, and I've put the location of the top binding position for lacosamide. and it's quite close to the residue of interest. It's not in the site of the Pfizer compounds, but there is a site that has high binding. The other place that lacosamide wants to bind is where you might expect, in the vestibule of the ion permeation pathway, and very close to these residues that can disrupt local anesthetic binding. Now, because of this, one of the things we needed to do is to ask, well, does lacosamide act here or act here? And when we mutated these residues so that we made it a non-local anesthetic binding, there was no lacosamide effects at all. So it appears that lacosamide definitely needs to get to these sites to be able to act. But what is the W1538 site doing? Well, the variant does not alter lidocaine action. So you can use other local anesthetic drugs, and it goes to that site in the ion permeation pathway, and the mutation from W to R absolutely does nothing to the action of lidocaine. So that site that appears to be able to attract lacosamide has nothing to do with the standard lidocaine local anesthetics. And as I said, we did 30 micromolar because it's clinically achievable, but we found that we could reconstitute an action of lacosamide if we increase the concentration. So in a strictly pharmacological way, we could overcome whatever reduction of activity of lacosamide to that variant by pushing the concentration. And so in slow activation, 30, and then 300, and in use dependence, 30, and then 300. So lacosamide does seem like it, to act, needs to get to the local anesthetic site, but we're still, curious about how could that site that's on the domain 4 Bolger-Sentian domain modulate things. So we did some other interesting things, which was since this is one of the sites that changed 1,7 to 1,3 or 1,3 to 1,7, we asked if that 1,3 specific compound could do anything to the 1,7, but it had this one of the 3 sites switched. And I think you can see that we're getting fast, slow, and use dependence responses to the 1,3 specific compound. So we're transferring a bit of the 1,3 pharmacology by having this variant, but trying to make lacosamide work better by making the analogous swap back. So instead of having arginine, it goes to tryptophan. We couldn't tell the difference. The lacosamide worked the same, either way. So whatever, we didn't try to do more, but there could be additional things. So our current hypothesis, and it's a hypothesis because we haven't directly tested any of this, is that the W1538 site is attracting lacosamide, and then in a sort of locally high concentration way allows it to access its site in the permeation pathway. And one of the things that we're considering is from these newer structures, it's very clear you can see these side fenestrations. And so we're interested to evaluate whether lacosamide comes in this way, and maybe that explains its slow kinetics of action. All right, so in summary, tabula rasa here, but in summary, we looked at the effect of lacosamide on variants that are associated with small fiber neuropathy, and we were able to get some correspondence between the pharmacological response in vitro and the clinical response of the patients. I talked about this particular variant. It's kind of interesting to us for mechanism of action. And when we looked at the responsiveness to lacosamide, the best responders actually enhanced fast inactivation responses, suggesting that they may be actually a better drug response than a patient with just wild type version of 1.7. And so the goal, maybe going forward, would be to throw other drug candidates on these variants and find one that would be the right one for those patients that carry that variant. And thus the aspirational pharmacogenomics in the title. And so I wanted to finish by acknowledging that a study like this cannot be done in isolation. It was part of a European group called PainNet, which the Maastricht is part of. And I really want to do a shout out to Julie LeBeau, who was a student with this program and did her doctorate work in our lab, in the lab of Dr. Waxman. And most of what I showed was in her thesis that she recently defended earlier this year. So she's a really up-and-coming scientist that if any of you want to contact her, I think she'd be great. And with that, I can take questions.
Thank you, Mark. That was outstanding. Right on time as well. Questions? No, it's a little early. I'll begin with the first one. So I have had my eye on lacosamide for a long time because it is also targeting the molecule I showed you yesterday, CRIM2. And there, they haven't been able to co-crystallize it or really find a docking site for it. With the channels, are you able to, do you have any knowledge of whether somebody's trying to figure out exactly where lacosamide's binding so to help further enable your studies?
I would say that, we have many things that we're interested in, and so we could perhaps try to, based on our in silico docking, try to figure out if that's actually where it's binding by mutating some of the other residues that are predicted, but we are not currently doing that, and I'm not certain if anyone else is doing it.
And then if no one has a question, a follow-up, the fenestration idea is very exciting. Do you think that so you basically suggested that that's perhaps the reason why you're getting that slow inactivation. So are there any plans to perhaps mutate some of those fenestration sites and see if that's affected?
And again, we have many projects ongoing, and I think Julie really wanted to do this, but she is not part of the lab right now. So we need a champion for this, and if the feedback here is that this is really important, maybe I can make the case that we should start this up again.
Fair enough. Lacosamide seems to be a very interesting, promising compound.
Yeah.
If there's no other questions, thank you very much, Mark.